Hi, I'm Dr. Scott Plevy from the University of North Carolina School of Medicine. In this part of the webcast, I'm going to tell you about Th17 and T regulatory cells in health and disease. This follows on to Dr. Calabrese's presentation that you just heard about two important T cell subsets, Th1 and Th2 cells, and their role in infectious and inflammatory diseases. Th17 and T regulatory, or Treg cells, as I'll call them from now on, are some of the hottest topics in human T cell immunology currently, and understanding their role in human disease is quite important and is going to really inform therapies in the future. The objectives of this part of the webcast are to describe the differentiation process for Th17 cells, identify the physiologic role of Th17 cells in the signature cytokine IL-17, as well as their pathologic role in autoimmune and chronic inflammatory diseases, describe the differentiation process for regulatory T cells, and recognize how Treg cells control inflammation through the production of anti-inflammatory cytokines. Many of the figures that I will show you in this talk come from Janeway's Immunobiology 8th edition. For this module of the webcast, I'm going to take T cell development from an evolutionary perspective. Why do we have T cells? And I want to put this in the context of why we developed Th17 cells and then T regulatory cells in terms of what they were normally meant to do, and then we could talk about what their functions may be in human inflammatory disease and why this may have come to pass. Well, to start this evolutionary adventure, this is a picture of Louis Pasteur, who, amongst his many accomplishments in helping describe microbial disease and vaccination strategies against microbial disease, he came up with what's known as the microbial theory of human disease. And what the microbial theory basically states is that one gets an infection because you're exposed to an infection. And it's really as simple as that. So if you look at any endemic infection or sporadic infection, you get it because you encounter a pathogenic organism. And the reason why if there's an influenza outbreak, some people get it and some people don't, is because the people who didn't get it weren't exposed, or even if they were exposed, they weren't exposed to enough of a virus in order to give this infection. However, what we learned is things weren't really as simple as that, and this led to the development of the genetic theory of infectious disease. In fact, if we look at the pedigree from Pasteur's family itself, you see that Pasteur had 3 children who actually died in the first decade of life from a febrile illness, and that's about all that was known back then. Let's go to the history of evolution and the godfather of evolutionary theory, Charles Darwin. If we look at Darwin's pedigree, you see that he also had 3 children who died of febrile illnesses very early in life. So these are 2 family pedigrees where one can assume that all of the children had identical environmental exposures, so you can't say that they all weren't exposed to the same pathogenic organisms, yet 3 people in each of these illustrious pedigrees died of what we now know as an infectious complication. So what this tells us right off the bat is that there's a genetic basis to infectious disease, and that the microbial theory wasn't in fact completely correct. So in fact, Pasteur didn't get the whole story. Prior to modern medical developments such as hygiene and antimicrobials, up to half of siblings in a family would die early in life from these febrile illnesses or fever. The microbial theory that illness was a matter of exposure, therefore, is only partially true and didn't account for intrafamilial heterogeneity in subjects exposed to the same microbial environment. So I'm going to ask and hopefully partially answer the question of what can the genetic predisposition to infectious diseases teach us about T cell development and function, because as I will show you, T cells, and in particular these T cell subsets, Th17, T regulatory regulatory cells, and as you heard from Dr. Calabrese, Th1 and Th2 have evolved for a reason, and the reason they evolved is to defend us against the environment and to control inappropriate immune responses. And with this theory that I'm laying out, I have to apologize to an investigator, John Laurent Casanova, who has been one of the instrumental researchers in describing some of these genetic defects in T-cell pathways that predispose to infections early in life. So with that, let's go into the first case. And this was Stephen D., who was first evaluated for chronic severe eczema at age 3. He actually developed seborrheic dermatitis and neonatal acne within the first few days of life. He also had recurrent skin infections with Staph aureus. And these skin infections were not typical of what you would expect to see with a bacterial infection of the skin. He would develop pustules, but they were not red or tender, and the term cold abscesses was coined to describe this. As you learned from your introduction to the immune response in this webcast, the immune response to an infection is usually characterized by redness, swelling, and tenderness, some of the hallmarks of the response to an infection. And this wasn't the case here, and that's going to tell us something important, as we'll get into. This patient also developed recurrent pneumonias, rhinosinusitis, and otitis media, and also developed mucocutaneous candidiasis. In terms of laboratory evaluation, the patient had a low white blood cell count of 1,040 with low numbers of absolute neutrophils in the white blood cell count. He had a markedly elevated IgE level. Sequencing of the transcription factor STAT3 gene, and I'll explain the importance of this later, revealed a missense mutation in the highly conserved DNA binding domain. And all of these clinical features, as well as these laboratory tests, supported a diagnosis of what's known as hyper-IgE syndrome. Well, since I took the approach of introducing you to T-cell subsets from an evolutionary perspective, to be fair and balanced, I have to get some scripture in. And hyper-IgE syndrome is also known as Job syndrome, after the biblical story of Job, of which the quote comes, "Why do the righteous suffer?" And Job actually took a challenge from the devil, where the devil said that he would renounce God if he suffered enough, and Job said, "No, I wouldn't." And what the devil did is inflicted him with these boils on his skin. So therefore, that's why the name Job syndrome applies to hyper-IgE syndrome, because these patients develop these cold abscesses or boils on their skin. Well, the hallmark immunologic finding in hyper-IgE syndrome is shown by our patient Stephen's blood analysis here, is the absence of a CD4 positive T-cell subset that makes the cytokine IL-17. What you're looking at here is a flow cytometric analysis from a healthy individual and a patient like Stephen with hyper-IgE syndrome. What you see in the lower right-hand corner, my right-hand corner of the normal flow cytometry on the left, is a proportion of CD4 positive T cells that co-stain for the cytokine interleukin 17. You see on the right side of the slide that this subset of T cells is completely absent, and this is the important immunologic defect in hyper-IgE syndrome, the absence of CD4 positive cells that make IL-17, which defines a Th17 subset. Before I discuss the specific factors involved in the development of Th17 cells and subsequently their function, let's do a bit of review of how T cells get activated in the first place, and this will go back to one of the earlier webcasts you heard from Dr. Looney before. A naive T cell first becomes activated through signal 1. Signal 1, you'll remember, is when the T cell receptor engages a specific antigen that's presented on an antigen-presenting cell, a dendritic cell or macrophage, most frequently in the context of a specific MHC molecule. In the case of activation of CD4 positive T cells, this is MHC class II. But signal 1 is not enough to get an activated effector T cell, and you need signal 2. This is the co-stimulatory signal. Signal 2 is generally mediated by B7 molecule expression on antigen-presenting cells interacting with T cell receptors such as CD28 and CTLA4. Signal 2, coupled with signal 1, then allows activated T cells to proliferate, survive, and perform effector functions. Now, when we talk about T cell subset differentiation, we invoke signal 3. Signal 3 is a soluble signal that's delivered to the T cell via cytokines that are made by antigen-presenting cells and other cells as well. Before I tell you about the signal that specifically directs differentiation of Th17 development, let's review what you heard from Dr. Calabrese. So for Th1 subset development, signal 3 are the soluble cytokines interleukin-12 and interferon gamma. These cytokines induce the signature Th1 transcription factor Tbet, which leads to the development of the Th1 subset. For Th2 cells, the soluble signal 3 is the cytokine IL-4, which then induces the signature transcription factor GATA3, which leads to stable subset development of Th2 cells. For the Th17 subset, the signal 3 are the cytokines TGF-beta and interleukin-6. More subsequent research have shown other cytokines are involved in signal 3 as well, including IL-1-beta and IL-21. As I'll tell you about in more detail, these cytokines work in concert to induce the signature transcription factor for Th17 development, ROR-gamma-T, which then allows the Th17 subset to develop, survive, proliferate, and perform their functions, which we'll discuss. Then afterwards, we'll talk about signal 3 for an important anti-inflammatory subset known as T regulatory cells. Well, with Th17 cells, there's an important other signal, which I will coin the phrase signal 4. And this is a signal that you're going to be familiar with and is going to be very important in the biotherapeutics of chronic inflammatory diseases, so you need to know about this. Interleukin-23 is a heterodimeric cytokine that shares a common P40 subunit with the cytokine IL-12 that you heard about as being a requisite factor for Th1 development, and IL-23 has a unique P19 subunit. IL-23 is a Th17 survival factor. What IL-23 will do is sustain the production and expression of the transcription factor FOXP3 in Th17 cells, leading to a sustained production of the signature Th17 cytokine, which I'll talk more about, interleukin-17. So again, this is downstream of differentiation, but an important sustenance factor, hence signal 4 for Th17 development. Well, I mentioned earlier in my case presentation of Stephen that there was a mutation in the gene for a transcription factor known as STAT3. Well, how does STAT3 fit into the development of Th17 cells, which are absent in the case of hyper-IgE syndrome? STAT3 is actually necessary for the expression of the Th17 signature transcription factor, ROR-gamma-T. Cytokines such as interleukin-6, which is one of the signal 3 for Th17 development, will bind to its cytokine receptor on T cells, leading to activation of the Janus kinases, or JAKs, which are cytoplasmic signaling molecules, and important targets to consider in human inflammatory diseases, with products well underway towards clinical development and use, which then activate the STAT proteins through phosphorylation events. The transcription factor STAT binds to the phosphorylated receptors and in turn is phosphorylated by these activated JAKs. The STAT proteins then form dimers that translocate into the nucleus and initiate transcription of this transcription factor, ROR-gamma-T. So without STAT3 activation, via IL-6, you actually block the production of ROR-gamma-T and you do not get development of Th17 cells. So that's how Th17 cells develop. What is their function in human disease and in health? So once again, to review what we talked about, if we have a naive T cell that receives signal 1 and signal 2, then signal 3, if it's comprised of TGF-beta and IL-6, as well as cytokines like IL-21 and IL-1 beta, will result in STAT3 phosphorylation, nuclear translocation, the expression of the signature transcription factor ROR gamma T, which then results in the expression and the production of the signature Th17 cytokines. The one I'll talk most about is IL-17, but there are others, including IL-21 and IL-22. IL-17 then has important biologic effects that explains the biology of Th17 in human health. In epithelial cells, such as keratinocytes and mucosal epithelium, such as in the lung and in the gut, IL-17 will result in the production of beta defensins and other antimicrobial peptides, which are involved in direct host defense against extracellular infections such as bacteria and extracellular fungi. Importantly, IL-17 will have effect on many other cell types that lead to the production of growth factors such as GCSF and GM-CSF that are involved in neutrophil development, and also multiple chemokines which will then direct neutrophils to the site of an infection. So Th17 cells in health are involved importantly in the defense of mucosal surfaces in the skin against extracellular infections by bacteria and fungi. And we'll get back to this because this certainly explains the infectious phenotype that's seen in hyper-IgE syndrome. Well, IL-17 is a pleiotropic cytokine and is the signature Th17 cytokine. There are actually multiple IL-17 family members that, just for simplicity reasons, I won't get into, which may have overlapping as well as unique functions. But IL-17 has effect on multiple cell types. I already mentioned it's going to lead to the expression of antimicrobial peptides. It's growth factors involved in neutrophil development, as well as chemokines involved in neutrophil recruitment to the sites of infections. It's involved in regulation of growth factors resulting in angiogenesis, factors such as matrix metalloproteinases that may lead to tissue remodeling, and inflammatory cytokines. So you could see where these effects on multiple cell types would have multiple synergistic effects in having protective role in the context of infections. Now, IL-17 also has an effect on osteoblasts, leading to osteoclastogenesis, chondrocytes, as well as the epithelial barrier, leading to decreased tight junction formation, which can contribute to the process of tissue remodeling and destruction as well. So although Th17 cells and IL-17 have multiple beneficial effects in the context of infection, too much of it, or in the wrong context, could lead to a deleterious role, as we see in chronic inflammatory diseases. And indeed, if we look at the expression of some of the signature Th17 pathway cytokines in chronic inflammatory diseases, such as IL-17 and the IL-23 subunits, which I said was a necessary survival factor for Th17 cells, we see that these cytokines are increased in inflammatory bowel disease, psoriasis, rheumatoid arthritis, and multiple sclerosis, suggesting that all of these cytokines may be viable targets for inhibition to achieve therapeutic benefit in the chronic inflammatory diseases that we treat on a daily basis. And other modules throughout this webcast and course will emphasize some of these points. The importance of IL-23 as a Th17 sustaining factor, as well as Th17 pathways in chronic inflammatory diseases, is probably best exemplified and proven by new genetic findings in these disorders. If we look at the IL-23 subunits, There have been multiple genetic polymorphisms that have been shown to predispose to psoriasis as well as inflammatory bowel disease. If we look at the IL-23 receptors, polymorphisms have shown to confer susceptibility as well as resistance to inflammatory bowel disease, psoriasis, as well as ankylosing spondylitis. If we look downstream of IL-23 signaling to some of the pathways that are important for Th17 development, we see genetic polymorphisms in some of the JAK molecules in inflammatory bowel disease, as well as psoriasis, as well as polymorphisms in STAT3, which we know is completely defective in hyper-IgE syndrome in inflammatory bowel disease. So genetics have told us about the importance of these pathways in chronic inflammatory diseases. So to summarize what Th17 cells are and what they do, these are an important component of the immune response against extracellular bacteria and fungi. Th17 cells, by virtue of secretion of the signature cytokine IL-17 and other cytokines, lead to neutrophil development, neutrophil recruitment to sites of infection, as well as the expression of antibacterial peptides. Th17 cells are defined by the expression of the signature transcription factor, ROR-gamma-T, which is driven by STAT3. Signals 3 and 4 are necessary for Th17 differentiation and sustenance. Signal 3 is prominently TGF-beta and IL-6. Other cytokines are involved. And signal 4 is the important cytokine, interleukin-23, which you'll be hearing much more about. Th17 makes the cells make the cytokine interleukin-17. There are several interleukin-17 family members which have overlapping but maybe unique functions as well. Other Th17 cytokines which are important in inflammatory responses that I didn't address just due to time and simplicity reasons are IL-21 and IL-22. Overactive or overabundant Th17 cells are present in chronic inflammatory diseases such as IBD, the inflammatory arthritides, and psoriasis. And these pathways have identified many new therapeutic targets in chronic inflammatory diseases, many of which have begun to infiltrate the clinic, including IL-23 blockade, IL-17 blockade, IL-21, IL-6 blockade. JAK inhibitors, which are in late-phase clinical trials, may have some effects through inhibiting Th17 cells, and inhibition of the signature Th17 transcription factor, ROR-gamma-T, is something of interest in the biopharmaceutical world as well. So let's get back to our patient, Stephen B., and what is the prologue here? Hyper-IgE syndrome is a single gene defect in the transcription factor STAT3. As I told you, STAT3 is necessary for expression of the signature Th17 transcription factor, ROR-gamma-T. Patients who have hyper-IgE syndrome lack Th17 cells and consequently develop extracellular and bacterial and fungal infections of the skin and at mucosal sites. So you may ask, it's called hyper-IgE syndrome. What's the explanation for increased IgE? This may be related to the lack of the Th17 cytokine IL-21, which I didn't discuss, but there may be other non-Th17 functions of the transcription factor STAT3 that explains this. The treatment for hyper-IgE syndrome is, for better or worse in this day and age, only antibiotic prophylaxis, but patients generally could do well if infections are prevented. Okay, we're going to move on to case 2, and we're going to go from the world that you heard about in Dr. Calabrese's presentation and so far in mine, from the world of inflammatory T-cell subsets, which have important roles in mediating host defense against infections, and if these pathways are inappropriately activated in the absence of infection, could result in chronic inflammatory diseases. And now we're going to go into the important world of a protective T-cell subset, which may prevent autoimmunity and dampen immune responses against self-antigens and innocuous antigens known as T regulatory cells. And to introduce you to this concept, let me also introduce you to case 2, the patient Billy S. And what you can see here is that Billy S. developed atopic dermatitis on his face very early in life. He developed atopic dermatitis shortly after birth, and then at 4 months of age, he developed intractable watery diarrhea, which was quite severe. This wasn't his only problem, and at 6 months of age, Billy developed type 1 diabetes, so autoimmune diabetes. On physical exam, he had diffuse axillary and cervical lymphadenopathy and an enlarged spleen. He actually required intravenous alimentation to keep up his nutritional status due to the severe diarrhea. An intestinal biopsy was obtained as part of the workup for this severe diarrhea, and what we're looking here is a small intestinal biopsy which shows this dense infiltrate. All of these blue cells you see between the intestinal crypts here are dense infiltrates of mainly inflammatory T cell populations. So this is an inflammatory process contributing to the severe diarrhea that is a cause of great debility in the case of Billy. Laboratory evaluation was performed, and as part of the workup for type 1 diabetes, the patient had anti-Gad65 antibodies and anti-pancreatic islet cell antibodies, which are two serologic hallmarks of an autoimmune process leading to type 1 diabetes. The patient also had elevated eosinophils and high IgE, so again, perhaps evidence of an increased T helper 2 response here, so there's multiple arms of the inflammatory immune process that seem to be out of whack here. The patient significantly had a brother who died in infancy with watery diarrhea and low platelets. Flow cytometry was performed on peripheral cells from Billy, and this revealed an absence of CD4 positive T cells that also have the T cell activation marker CD25, which is the interleukin 2 receptor, and it also revealed an absence of CD4 positive T cells which express the intracellular transcription factor FOXP3, which I'll get back to. Sequencing of the FOXP3 gene revealed a missense mutation which led to lack of expression. Subsequently, Billy developed thrombocytopenia and antiplatelet antibodies. So now the patient has autoimmune thrombocytopenia in addition to inflammatory enteropathy and type 1 autoimmune diabetes. So what does Billy has? Billy has something known as IPEX disease or IPEX syndrome. IPEX stands for immune dysregulation, polyendocrinopathy, because the autoimmune process affects multiple endocrine glands, and enteropathy, which is autoimmune in nature also, which is X-linked. In IPEX disease, there is a failure of peripheral tolerance to self-antigens due to defective production of a subset of T cells known as regulatory T cells or Treg cells. Treg cells are CD4 positive and CD25 positive, so activated T cell populations that express the transcription factor FOXP3. How do Treg cells develop? And there's a signal 3 involved in T regulatory cell development that I'm illustrating on the left side of this slide. Signal 3 occurs for T regulatory development, importantly, in normal homeostasis, so in the absence of an infection. In the absence of an infection, professional antigen-presenting cells, like dendritic cells, make this predominantly the cytokine TGF-beta and very little of the cytokine interleukin 6. And in a milieu where dendritic cells are making high levels of TGF-beta and low levels of interleukin 6, as well as interleukin 23, a naive T cell that receives signal 1 and signal 2, these conditions are permissive for the development of CD4 positive T cells that are activated to express FOXP3 and show a regulatory phenotype. The regulatory phenotype, as I'll discuss in more detail, being that these FOXP3 cells, regulatory T cells, can inhibit inflammatory T cell populations, including Th1, Th2, and Th17 cells. Now, the development of T regulatory cells hangs in a precarious balance. I told you earlier in this talk that TGF-beta is one of the requisite factors for the development of inflammatory Th17 cells. Now, if TGF-beta is produced for example, is illustrated on the right side of this slide. In the setting of an infection, dendritic cells and other cells not only make TGF-beta, but they also make IL-6 and IL-23. Under this milieu, naive T cells that receive signal 1 and signal 2 will actually go on to express ROR gamma T and become Th17 cells, which then survive by virtue of the fact that IL-23 is being produced, signal 4, as we discussed before. So you could see here whether there is a potent anti-inflammatory cell, Treg cells, or a potent pro-inflammatory cells, Th17 cells, hangs on the precarious balance of whether interleukin-6 is produced. If you get TGF-beta without interleukin-6, which occurs under normal conditions of homeostasis in health, you get T regulatory cells, but if an infection enters the picture and IL-6 is made, then you get this inflammatory T cell subset, Th17 cells, which develops to eradicate these infections. There are two kinds of Treg cells which have become known as inducible and natural Tregs. T cells specific for self-antigens, antigens expressed on cells of our body, are recognized in the thymus and become natural regulatory T cells. So these cells are educated against self-antigen in the thymus under the influence of TGF-beta, and then can home out to the periphery where they will stop autoreactive T cells from developing and prevent autoimmune attack. However, natural T cells develop in the periphery. T cells specific for self are in fact innocuous antigens, such as antigens presented by commensal microbiota in the gut or in fact on the skin, or dietary antigens that aren't meant to elicit immune response, such as will be encountered in the gut all of the time. These T are recognized in the presence of TGF-beta in non-inflammatory circumstances, and in the presence of TGF-beta, cells can be induced in the periphery, mainly in the gut and in lymph nodes, to become natural Treg cells, which will then prevent the development of responses to both self-antigens as well as innocuous antigens, such as the microbiota that normally colonize our gut, our lungs, our skin, as well as dietary antigens. T regulatory cells perform many of their anti-inflammatory functions due to the cytokines that they produce. And there's two very important cytokines produced by T regulatory cells that explain many of their anti-inflammatory functions. And these are the cytokines interleukin-10 and TGF-beta. IL-10 and TGF-beta are known to inhibit either self-reactive T cells or inflammatory T cells, Th1, Th17, Th2, via direct effects on these T cell populations. What I'm showing you on this slide is a classic immunologic assay for T regulatory function, and this is a suppressor assay. And what happens here is that CD4, CD25 positive T regulatory cells that express FOXP3 are titrated in at increasing numbers to effector T cell populations, Th17 or Th1 cells, for instance. And what is shown is that T regulatory cells can dose-dependently suppress proliferation as well as cytokine production by effector T cells. And what's interesting and important is they do this in an antigen-independent manner. So T regulatory cells from any source can suppress antigen-specific T cell effector responses to any antigen given. So there's no antigen specificity for T regulatory cells' ability to suppress inflammatory T cell responses. Well, T regulatory cells play a particularly important role in the gut, and as a gastroenterologist who study the inflammatory bowel diseases, I think that gut biology is probably the best example of how T regulatory cells may function in health and disease. What I'm showing you here is a photomicrograph of a normal colonic intestinal biopsy. If you look at the top side of the slide, you see the gastrointestinal lumen, and it's important to keep in mind that this lumen is colonized by an incredibly large biomass of intestinal microbes, including bacteria and other microbes. In fact, the number of microbes in the gut lumen outnumber the cells in our human body by 10 to 100 to 1. So there's this incredibly complex biomass of microbes that, if exposed to the immune system in any other organ in the body, is going to elicit a profound effector inflammatory T cell response and cause multiple human infectious diseases and inflammation. In the gut, though, the normal response is homeostasis. Inflammation does not result. So what I call this state is controlled or physiologic inflammation. And this is illustrated by these number of blue cells you see in the lamina propria of the gut that live in between the single layer of epithelial cells which invaginate into the lamina propria, known as the epithelial crypts. Each of these blue cells represent a cell of the immune system, and over 50% of them are T cells. If this was any other organ in the human body, what you would be looking at is chronic inflammation. But in the setting of the gut, this is physiologic inflammation. And in fact, what we know is most of these cells that we're looking at are CD4 positive, CD25 positive, and express FOXP3. And these cells are there to prevent the immune system from responding to innocuous antigens, such as components of the bacteria that live in the gut that normally leak through the intestinal epithelium in all of us on a minute-to-minute basis. You would not want these T cells to respond to every bacterial antigen that they see, because most of the bacteria living in our gut lumen are innocuous, and in fact it's a true symbiotic relationship that we're dealing with. Likewise, when we eat a hamburger, we don't want our immune system to react to any of the dietary antigens that they're exposed to once they're absorbed through the epithelial cells in the gut. So these T regulatory cells are there also to calm down immune responses to dietary action. So this is the default pathway in the gut. Now, in a disease like inflammatory bowel disease, and in our patient who has significant enteropathy, there may be a defect in T regulation. And what has been shown in mouse models of inflammatory bowel disease is that T regulatory cells may actually prevent inflammatory bowel disease. So in mouse models, and probably in some forms of human inflammatory bowel disease, this results from autoreactive T cells that are present in the lamina propria that don't belong there. The disease actually in mice can be treated by adoptive transfer of CD4 positive, CD25 positive T regulatory cells, which then home to mesenteric lymph nodes, which are the lymph nodes of the gut, and then go to the colon. These T regulatory cells then proliferate and inhibit the pathogenic effector Th1 and Th17 responses that we see in the gut. So after inflammation resolves, these T regulatory cells remain in clusters with dendritic cells as well as pathogenic effector T cells, and this actually shows you that they're there to dampen the function of these effector T cells. Now, in normal homeostasis, there's also these effector T cells in the gut that are ready to respond in the setting of infection. So going back to what we talked about already, if there's an enteric infection, the dendritic cells may start making IL-6. We're then going to see these Th17 cells emerge. We're going to get IL-23. They're going to proliferate, and they're going to be able to do the job against infections. But in the normal state, these T regulatory cells are making sure that these Th17 cells are not activated to innocuous stimuli from the environment, such as the enteric microbiota and dietary antigens. So in summary, what are CD4 positive Treg cells? These are the cells that are responsible for why we don't react to self-antigens or innocuous environmental antigens. They express the cell surface molecule CD25, which is the interleukin-2 receptor, which causes T cells to proliferate, and they express the signature transcription factor FOXP3. Signal 3 for Treg development is TGF-beta in the absence of IL-6. Again, in the setting of infections, IL-6 is going to be induced, and this pathway is going to be pushed towards Th17 cell development. Natural T regulatory cells are produced in the thymus and protect against autoimmunity to self-antigens. Inducible T regulatory cells to self as well as environmental antigens differentiate in the periphery. T regulatory cells subserve many of their functions through production of the potent anti-inflammatory cytokines TGF-beta and interleukin-10. A very hot topic in immunology is how can we harness the power of these T regulatory cells to treat various human diseases. There are many strategies that are being tested in preclinical models as well as in the clinic to actually induce T regulatory cells in autoimmune and chronic inflammatory diseases. And there's actually strategies that have already hit the clinic to look at the opposite side of this equation. And one of the reasons that certain cancers, such as melanoma, that are immunologically responsive may grow and proliferate out of control is because there's a strong T regulatory response that inhibits effector T cell responses from recognizing and destroying the cancer. So there is a certain strategy, a monoclonal antibody to the cell surface marker CTLA4, which is a co-stimulatory marker, the signal 2 again, which is expressed on T cells but is enriched on T regulatory cells. So anti-CTLA4 antibodies has been shown to inhibit T regulatory cells, allowing an effector T cell response against melanomas. And interestingly, by inhibiting T regulatory cells, one of the side effects and consequences of anti-CTLA4 therapy in melanoma is the development of autoimmunity. So this makes sense. Well, what about a prologue for our patient, Billy S.? IPEX disease is caused by a single gene mutation in the transcription factor FOXP3, resulting in defective T regulatory cell development and severe systemic or autoimmunity. Billy S. was initially treated with immunosuppressive therapy, including cyclosporin and tacrolimus, to inhibit activated T cells. The diarrhea, glucose control, platelet count, and eczema all improved markedly. However, after 6 months, all of his symptoms recurred and he became refractory to this medical therapy. Billy subsequently underwent a bone marrow transplant from his 5-year-old HLA-identical sister. A year after transplant, Billy remained symptom-free. So by replacing the hematopoietic component with bone marrow from Billy's sister, Billy now has T-cell precursors that express FOXP3, which can be educated in the thymus as well as the periphery to become T regulatory cells and suppress this out-of-control autoimmune response. So bone marrow transplantation is a potential cure for IPEX disease. Okay, to wrap up my presentation, I started with evolution. Let's get back to evolution. And the Human Genome Project has opened new ideas and new thoughts about the pathogenesis of chronic inflammatory diseases. The disease that has consumed my clinical practice and research interest has been the human inflammatory bowel diseases, Crohn's disease and ulcerative colitis. And we're now well over 100 disease susceptibility loci that have been described. What's really interesting, and I highlighted earlier in my talk, over half of these loci are actually shared with other chronic inflammatory and autoimmune diseases, including psoriasis, the spondyloarthropathies, multiple sclerosis, rheumatoid arthritis, and type 1 diabetes. Well, evolutionarily, the story even gets more interesting. There are several loci shared amongst patients with IBD and other immune-mediated diseases that actually confer susceptibility to the development of systemic leprosy, which is an intracellular Th1 type infection with the bacteria Mycobacteria leprae. There's another important overlap with a whole series of genes that confer Mendelian susceptibility to mycobacterial disease with other rarer forms of mycobacteria. These are your single gene infectious loci that predispose to the development in early childhood of rare infections. Sound familiar from earlier in my talk? It should. So this leads us to think about the diseases we treat in a whole new evolutionary context. And it is not surprising that many of these overlapping genes we see between inflammatory bowel disease, other chronic inflammatory diseases, and susceptibility to mycobacterial infections are well placed within the Th1 and Th17 pathway, including genes in the IL-12 and IL-23 pathway, the JAK pathway, STAT3, interferon gamma signaling. So this allows us to speculate that the evolution of inflammatory T-cell responses and regulatory T-cell responses may actually serve a purpose. And we can ask the question now whether genes that predispose to autoimmunity and chronic inflammation actually at some level confer a survival advantage through protection against infections in early childhood. Conversely, an exuberant Th1 or Th17 response or diminished T regulatory activity could have possibly been selected for by endemic infectious organisms and populations. However, under modern environmental pressures in the context of hygiene and antimicrobial agents, other environmental pressures could lead to other problems such as autoimmunity and chronic inflammation. So this balance between inflammatory and regulatory subsets probably has an evolutionary purpose in terms of defending the host against infections, but in the modern environment and in the setting of hygiene and antimicrobials, these exuberant inflammatory responses which are genetically predisposed actually may result in chronic inflammatory diseases that we treat in the clinic. Thanks for your attention.